Microorganisms do not always make us ill. The correct composition of bacteria in our intestines can make us healthier and even heal illness. Scientists will now program bacteria to produce medicines that can make us healthy when we are sick.
Our goal is to create a living medicine that can respond to what happens in the body and provide a more tailored delivery of pharmaceutically active molecules.
With current medicine, dosing is a big challenge.
Medicine, the way we know it today, is based on either small molecules or proteins that we inject or eat as a pill at a certain number of times per day. And that gives certain limitations in terms of the way that we can deliver these therapeutically active molecules.
The distribution of the medicine over time varies a lot and does not result in optimal effects.
We think that by engineering bacteria to respond to signals from the body and to produce these pharmaceutically active molecules on demand, that we can address these limitations of current medicines and deliver dosing regimens that are much better suited to treat diseases.
The idea behind self-based medicines is to treat a disease in a similar way to how the body would do it.
The way the body would respond to a disease, for instance diabetes, is that it would produce insulin in response to certain signals in the body, and that would lower the blood sugar. We can achieve a similar thing with bacteria because we can engineer them in principle to respond to those same signals and produce those same molecules.
Bacterial self-therapies can potentially influence a very broad range of diseases. The project will test 3 concepts.
The first one is that we can produce a compound that is pharmaceutically active. The second one is that we can remove a compound that might be harmful to the body. And the third concept is that we can engineer the composition of the bacterial flora in the host that might be associated with a particular disease.
Using self-based medicines requires strategies to control the medicines so they do not spread to other people.
Over the past decades, we have really built up competencies to engineer very sophisticated control systems in bacteria. And that would allow us to, in a variety of different ways, to control that a bacterium persists only in the patients that are treated and only for the time period that we would like them to persist for.
However, the greatest challenge in developing bacterial self-therapies is the interactions within the complex bacterial communities.
Currently, there is relatively little information about how engineered bacteria will interact with a bigger community of bacteria. And that's going to be a major thing that we will study in this project. And we will aim to develop strategies that allow us to override, in some ways, these interactions so that we can control specifically the abundance of our engineered bacterium.
Of course, the researchers dream of developing a new treatment for some life-threatening diseases. However, the long-term perspectives are even more important.
A successful outcome of this project would be that we have developed a synthetic biology framework for how we can engineer bacterial cell therapies, how we can contain bacterial cell therapies, and how we can use bacterial cell therapies to affect the human host. If we can achieve all of those things, I think we've had a successful project.